σας. Σας καλωσορίζω σε αυτό το σεμινάριο, όπου έχουμε τη χαρά να έχουμε μαζί μας τον Αναστάση Περράκη από το Cancer Institute της Ολλανδίας, ο οποίος θα σας μιλήσει για αυτό το θέμα, το οποίο θα δούμε παρακάτω. Τώρα, όσον αφορά τον Αναστάση, είμαι σίγουρος ότι πολλοί από εσάς τον έχετε δει επανειλημμένος. Η σύνδεση του Αναστάση με τον χώρο εδώ πέρα, το IMBB και της Κρήτης, αρχίζει από πάρα πολύ παλιά. Έτσι, εδώ βλέπετε το 1990, όταν είχαμε οργανώσει ένα course για protein design, όσο και αν φαίνεται περίπου αυτός εδώ είμαι εγώ, τότε νέος επίκουρος καθηγητής, έτσι, και εδώ πέρα αυτός εδώ είναι ο Αναστάσης, έτσι, ο Ηλιόπουλος είναι αυτός. Συγγνώμη, δικαιολογούμε πάντως ή λίγο δεξιότερα. Εντάξει, τότε συγγνώμη, υπάρχει μια σειρά από φωτογραφίες, άντρες να κάνουν guess από τις πλάτες, οπότε εδώ έπεσα έξω, έτσι. Πάντως όλοι αυτοί έχουν γίνει καθηγητές στο μεταξύ. Εντάξει, εγώ παρέμεινα. Τώρα, ας πούμε μερικά πράγματα για την επαγγελματική εξέλιξη του Τάσου, μετά από αυτή την φωτογραφία που δεν το είδατε. Ο Τάσος πήγε στο Αμβούργο, στην ομάδα του Keith Wilson, προερχόμενος βέβαια από το Πανεπιστήμιο της Αθήνας, όπου δούλεψε με διάφορα χιτινογητικά ένζημα, δημοσίευσαν διάφορες εργασίες οι οποίες έχουν τύχει μεγάλης απήχησης, και μετά την ομάδα του Keith Wilson πήγε στην Ολλανδία, σε αυτό το NKI, το Ινστιτούτο του Καρκίνου της Ολλανδίας, και δούλεψε εκεί πέρα σε θέματα που αφορούν DNA mismatch recognition και γενικώς πρωτεΐνες που συνδέονται με το DNA, και στη συνέχεια και παράλληλα επίσης ανέπτυξε software το οποίο χρησιμοποιείται ακόμα τώρα ευρύτατα στην κρυσταλλογραφία για την ερμηνεία των κρυσταλλογραφικών δεδομένων, και αυτό έχει δημοσιευτεί σε περιοδικά τα οποία επίσης έχουν πάρει χιλιάδες citations. Μετά από την Ολλανδία, ο Αναστάσης πήγε στο EMBL της Γκρενόβου, στην ομάδα του Steve Κούσακ, και εκεί πέρα βοήθησε στην ανάπτυξη του beamline στην Γκρενόβου, και το 2000 επέστρεψε στην Ολλανδία σαν group leader πλέον, όπου συνέχισε τη δουλειά με τις πρωτεΐνες που συνδέονται με DNA και σχετίζονται με διάφορους καρκίνους, ορισμένες από αυτές σχετίζονται με γενετικές τροποποιήσεις κτλ. Ενώ έχει συνεχίσει παράλληλα και την ανάπτυξη software για το modeling διαφόρων πρωτεϊνών. Ο Αναστάσης είχε πολύ ενεργό ρόλο σε διάφορα μεγάλα διεθνή προγράμματα, όπως είναι σε θέματα structural genomics, την πρωτοβουλία αυτή της Spine, και τα νεότερα χρόνια εκεί πέρα είχαμε και συχνές επαφές, ασχολήθηκε και με προγράμματα τα οποία εξασφαλίζουν την πρόσβαση σε μεγάλες εγκαταστάσεις συγκροτωνίων σε όλη την Ευρώπη, και νομίζω όταν θα έρθει η ώρα της ομιλίας του θα σας πει πέρα από το επιστημονικό και μερικά θέματα που σχετίζονται με αυτά τα οποία παρουσιάζουν ενδιαφέρον για όλους. Είναι έντυπος σε διάφορα περιοδικά, έχει υπηρετήσει σε διάφορες επιτροπές για διεθνείς χρηματοδοτήσεις, όπως επίσης υπήρξε εξωτερικός εξεταστής διδακτορικών σε διάφορες χώρες, μεταξύ των οποίων και στην Ελλάδα. Οπότε, με αυτή τη σύντομη εισαγωγή, είναι χαρά μας που έχουμε τον Αναστάση εδώ, και θα ήθελα να τον παρακαλέσω να μας παρουσιάσει το θέμα.
ευχαριστώ για την λεπτομερή εισαγωγή. Ευχαριστώ που ήρθατε. Υπάρχει μια τάση, όποτε μιλάω στο Ηράκλειο, μιλάω σε πολύ μεγάλες αίθουσες που δεν έχουν απαραίτητα πάρα πολύ κόσμο. Και αυτό δεν το λέω μόνο για το είτε, όσοι κατάλαβαν. Λοιπόν, θα χωρίσω την ομιλία μου σε δύο κομμάτια.
Επειδή έχουμε μία ελληνόφωνο στο αγροτή.
Ναι, και είπα η επόμενη φράση είναι ότι θα γυρίσω και στα αγγλικά. Λοιπόν, as Michalis told you, I work at NTI at the Department of Biochemistry. NTI has a peculiarity. It's an integrated research center and a hospital. So we're a real hospital with real patients. We specialize in very specific forms of cancer, namely lung, melanoma, gynecological cancers, and male cancers. That's actually the biggest part of the organization, with over 1,000 employees. And they're really not just in the same campus, but really in the same building. There's about 10 basic research departments and 5 clinical research departments. Over the last 10 years, a very big thing in the Institute, trying to move towards multidisciplinary research, is actually having several facilities, allowing us to go really, allowing, for example, for my lab to develop a mouse model, and people that do animal studies to work on proteins, when the right questions are there, in a facility rather than necessarily in a collaborative manner. We are in the old department out. I guess that's why we're in the top floor, actually to have better cooling for our X-rays. We're 3 groups there, we're 4 groups there. It's Titia Sixman, who's the head of the department, who works on DNA repair and signaling. Tijn Bloemelkamp, who actually is the gold medal winner from EMBO a couple of years ago, who is doing quite a bit of functional genomics, but with a very strong aspect on looking also at function and following up his projects. And then it's a protein facility, which I also actually scientific supervise on my group. So we have several technologies available, going from protein expression to high throughput crystallization. We can now these days, we don't have a microscope, but we can prepare our cryo-EM samples and use the National Center, because I guess most of you are aware that over the last couple of years, the development of direct detectors technology for electron microscopes have allowed routine structure solution of big complexes down to 2.5 angstrom resolution, which is directly comparable to quality with crystallography, actually. A very strong aspect of the lab is our biophysics laboratory that we have in a very consolidated way several equipment that the final aim is to measure the interaction of proteins, the stability of proteins, and anything to do with proteins. And that's where INEXT counts, which is an EU program that I'm going to spend like 5 minutes telling you about. INEXT stands for Integrated NMR-EM X-rays for Translational Research. And it was a response to the call from the Commission a couple of years back, a bit more than a couple of years back actually, that infrastructures that are concerned with structural biology should group together and try to attract new communities of users, and in particularly scientists with backgrounds other than structural biologists. So INEXT, besides making sure that Michalis and Kyriakos, for example, have their access to their X-rays, the real challenge for us is to attract people that just want to do structural biology and they don't have the equipment or they don't have the knowledge, but they have the clearly defined scientific questions for that. Actually, if you want to know more about it, besides reading quickly through that, there's a few brochures outside the room, and come and talk to me. But very briefly, it's NMR Center, synchrotron CMs. I'm actually the acting coordinator most of the time for that, but we also host in our lab a modality to look for protein-protein interactions. And that's the infrastructure we offer is our biophysics lab. And all this actually is also integrated with Instruct, which is another, so I'm doing a bit of the sales piece for facilities now. It's a European infrastructure initiative. More of you, I guess, are familiar with Elixir, for example. Instruct is the equivalent of Elixir for structural biology. And there's quite a few participating countries. Greece for now is an observer status. But still, like, we offer infrastructure facilities, and that's done in collaboration with INEXT. Something particular is like, again, you can mail in your samples for analysis. You can go for a week or two to be guided through crystallizing a protein and solving a structure. You can check if your sample is amenable to electron microscopy. Very soon we'll have availability for fragment screening, like basically the initial steps for structure-based drug discovery. And we also participate together with Corbel and Eurobioimaging to offer access to advanced light imaging with techniques that directly address scientific questions that are relevant to structural biology, at least. Something that I particularly want to advertise on INEXT is this modality. So it's the Structures for the People modality. We call it structural audit. That's we're trying to catch attention with this poster. And the idea is that we want to offer to biochemists and cell biologists and whoever thinks that for their research they have a problem that interests them for structure, and perhaps they made a protein, and the protein, is enzymatically active, and then they go like, well, it would be interesting to solve a structure for that and that reason, but we don't know how to start. So what you can do actually is that you can put a few microliters of your sample to the mail. We pay for the FedEx to come at NKI, and we go through a very extensive analysis of the properties of that protein. We try to crystallize it. We collect small angle scattering data for you. If it's big enough, we try electron microscopy, and in a couple of weeks you get your sample back with all the results. You have no obligation, you don't need to pay. What you can do is that you can put these results in your next grant, because they might help you to get funded. You can establish a collaboration with your local crystallographer. We thought it's very attractive. Apparently it's not oversubscribed, to put it like that. So we also try, we try to attract different audiences. Some like this poster. There was some disagreement that if it's a social argument or rather than a financial argument, but they're for free as well, these structures. Okay, so 5 minutes down the line, end of sales pitch. So what we do in my lab, we do structures quite a lot, or actually we used to do structures. I come from X-ray crystallography, but I think as you'll see in my main talk a bit, the personality of my group, if you want, has changed quite a bit over the years. Since I am a crystallographer, and Michalis mentioned our warp, I'll try to explain more or less what that is. So the idea is that when you do a crystallographic experiment and you solve the infamous phase problem, which I'll not bother you about, you end up with this chicken wire, this map, and we call it the electron density map. So what this shows you is that inside the contour, it's very likely that you have protein atoms, you have the protein chain and so on. But still, what crystallographers had to do was to build these models, and you would spend a few days, or if you're a PhD student, you would spend the nights, because during the day they would be your supervisor there, to build the models in these very expensive machines, actually you could get awful nightmares swimming in electron density, trapped in an Argentine side chain. I don't know if Kyriakos had it perhaps as well. I did. So at some point, together with my friend and collaborator at Hamburg, Victor Lansing, we had this idea that basically was like, okay, we can take this electron density map and throw in it these blue, we call them pseudo-atoms. And this was at the time a more efficient way to describe this very complicated shape in a slightly different way with less data, with less parameters. You see, like, this is just XY coordinates of these atoms. It's not this complicated three-dimensional probability function. And what that allowed us was to use pattern recognition algorithms that were based a lot around the reduction of the parameter space to build automatically with zero user intervention molecular models. And by some tricks that we would iterate, we could at the end complete these models. And this became actually very popular in the late 90s because we were the first ones that could do that automatically for something at the time 2/3 of the structures that were solved. And since structural genomics were taking off at the time, and people were trying to scale up things, and this was by far the most time-consuming step, this became very successful. Over the years, then other people did it, and admittedly a couple of people did it better than us, also not at high resolution. But I will not argue about that. My research interest shifted to a different problem. Actually, I was scooped with this structure at 2004, 4 years about starting my group. That structure was one of the panels in a Cell paper, and without my name on it. That almost costed me my career because all of my group was working on mitosis since that time, and this protein is the polo-like. It's a small bit of the polo-like kinase 1. It's usually described as the choreographer of the cell cycle. And what you see here is this is a bit to the right. It's a small bit of polokinase, and to the left is a small bit of a peptide that is binding to polokinase. So now you see some interesting features chemically. For example, this oxygen and this oxygen are looking towards each other. And there is this red bit of density there, which means that this should not be there. That's red is bad in crystallography. And there's also this green density over here saying that there should be something there. Also, part of the discussion even of this paper was like, this is like the affinity, the biochemistry shows that this is a very strong affinity for recognizing this peptide, but it's not clear from the structure. Well, it becomes very clear from the structure if what you do is that you flip the oxygen the other way around and you correct the structure, there is no residual green or red density anymore, and you realize that you get here two excellent hydrogen bonds. So now you can spend as much time as you want arguing that these people were in a hurry, that they didn't know what they're doing, that it was a mistake of somebody and so on, but the fact is that there are a lot of errors in the protein data bank. The protein data bank, the PDB as most of you know, is an archive. A crystallographer does a structure, puts in the protein data bank. The protein data bank is allowed to tell you, well, maybe that's not necessarily correct, but it's a scientific and technical decision, and it's your responsibility what you put there. In the past, in the 70s and 80s, people did not have the computing power to do things properly. And more recently, people do not afford the time, or quite frankly, a lot of people that are doing structures had what one could call suboptimal training. I want to say a bit more on that of how bad the mistakes were, because my postdoc Robbie Yoston that was doing this work, at some point he had to present to this staff meetings that is called Cancer Institute. And we said, he asked like, can I convince the medical doctors that this has something to do with cancer? And I said, well, what will they know? All they know is Herceptin. It's the antibody used for some forms of the HER2 plus breast cancers. And he said, okay, let's look at the structure of HER2, the receptor extracellular domain with Herceptin. That's, Herceptin is an antibody that's used for the last 10 years almost in the clinic. And again, for Herceptin, there is these things, it's raised on the paper describing this, that the protein-protein interface is quite unusual because there's not many interactions that would justify how well this antibody works. And indeed, now what you see here is just a small part of this interface over here. You see indeed, like in this area, there's just one hydrogen bond. And what do we do now? We don't do anything. It's our software that goes through that and tries to look at the data and redo the interpretation of that model. The picture that you get is like that. Just in this small part, there's not one hydrogen bond, but there is a whole network of hydrogen bonds. There's even a couple that I am not displaying there that actually explain very well how Herceptin works. And if you want to engineer this antibody, which people are doing, you would rather be having this structure rather than that structure. Okay, so that's all I wanted to tell you about methods. I'll go before again. I will still spend 2-3 minutes showing one of the projects that I'm not going to talk about. And this is this protein here called autotaxin. So autotaxin binds lipids, and we published a lot about how it makes LPA, which is a signaling lipid. What we have found out over a couple of years ago, that next to the lipid, there was this molecule, which after a long time, we realized that, because we didn't know that, we didn't know what was bound, we realized that this is a steroid. And this is, and exactly the class of the steroid is called a bile salt. And what we found out is that our protein that we were expressing in mammalian cells, and we were purifying for structural studies for over 10 years, for all these 10 years, it always had this molecule there, which was a steroid. Interestingly, we only were able to find that when we moved from below 2 angstrom resolution. By mistake, we did the structure at 1.4 angstrom, because some people ask sometimes, why do you want high resolution structures? Well, because sometimes you get surprises and sometimes you get to learn new things. And there was one aspect, we published that work earlier this year, because the fact that you have this interplay on steroids regulating the activity of autotaxin in producing the signaling lipid is very much related to at least a couple of diseases. For sure it's related to pruritus, which is hepatic disease basically, but it's also likely implicating some neuronal disorders. The interesting bit about it, and this usually does not happen, but I'm very happy it happened to us, is that we went to our chemist collaborators in Glasgow last December. Lovely place to visit in December. And we asked them, we sold them this idea and with this data before publishing and said, like, could we make an inhibitor that actually combines a lipid and a steroid? Because this could be really very specific. And indeed, they started drawing and they said, okay, what we'll do is that we have our steroid here, and actually we have this inhibitor from Pfizer that works pretty well. But the problem of this inhibitor is this group over here. This is a very reactive group, and you don't want it in patients because it reacts with a lot of things. So let's take this and put it to that. Let's make this inhibitor. They said this is very easy. The chemistry is trivial. I don't understand it. But the fact is that 3 months later they came with some compounds. In a month, really, we had some activity data. We solved a couple of structures that showed us how these bind. And by June, we had new compounds that were designed based on the structure. And in August, we had all the data on activity, structure, and kinetics. This is the structure of this hybrid inhibitor that you see the bile acid and the old Pfizer molecule, as we called it. And the interesting thing with that, it's something very peculiar. The active site is here. So this inhibitor is not interfering at all with the active site of the protein. It's only interfering with the binding of the lipid and the binding of the steroid. So the advantage of that is that because it only interferes about binding, and active site chemistry, a catalytic threonine acting as a nucleophile, bimetallic zinc atoms, this is classical phosphatases. and you want selectivity. And this is very selective because it doesn't bother to go at all to the active site. And what we could also, that is not yesterday, that was Wednesday, we could characterize the inhibitor. We see that it works in cells, that if we look at downstream signaling from autotaxin, you see large induction of signaling with an IC50 of around 20 to 30 nanomolar. And most importantly, I'm sorry this is so small, but when you inject this compound to mice, this is what happens when you inject the vehicle. This famous Pfizer inhibitor puts the things to half, but it's quite toxic. It's only, the idea is now phase 2 trials for patients that have either cancer or fibrotic disease in the very last steps that you don't really care about toxicity. But this drug that hopefully is not toxic, we're waiting for the mouse experiments, is as active as the Pfizer compound without going to the active site. So my postdoc was instructed to have the paper ready for Wednesday. I don't think this bit will work. I will not show this because I've already spent 20 minutes. And finally, it's my talk. So I need to take a breath at this point. So that's the science bit and not what I'm doing. So working on mitosis is possibly the longest living project in my lab. And we've been quite remarkably and successful on making the contributions that we would have liked to do in the field. And one of the problems that we work almost for 8 years is MPS1, the monopolar spindle 1. So what is this protein important for? Is to avoid exactly this situation here. So here you see like there has been a mitotic spindle formed, and we are in anaphase. These are the chromosomes. They're being pulled to the future daughter cells, and you see this little chromosome left behind. So this chromosome had the attachment tissue. It has not been attached to microtubules, and if the cell was functioning properly, what it should have done is that it shouldn't have left this chromosome behind. And if you look now to mammalian cell dividing, you see here the spindle in green, all the chromosomes are in the middle here, and I'll play this video again because you see that what happens is that the cell spends quite some time, no, that's the wrong movie, I'm sorry, it spends quite some time making sure that all the chromosomes are aligned here. You see, that's quite some time. And then it pulls them apart, when all the chromosomes have been attached bipolarly. What happens often in a cancer cell, and it's over here, try to have a look at this video now. Again, chromosomes go to the middle. Look at this guy here. You see? Everything happened quickly. The cell did not spend enough time to check because it had a defective mitotic checkpoint. And actually, this is a cell that has an MPS1 knockdown, and it exits mitotic very quickly without checking attachment. This is a situation that leads to different kinds of polyploidies and aneuploidies, which are very common in cancer. Something more than 80% of solid tumors have this problem, and naturally drugs that target the mitotic checkpoint proteins are a very big promise and soon beyond the promise for the clinic. So two of the situations that occur in chromosomes is that you can have this situation over here that you can see like this kinetochore is attached to tubulin, that's the tubulin, that's the kinetochore in the middle of the chromosome, and this is not unattached. You can have other issues as well. That's another big interest of our labs, is that you have the same microtubules coming from the same side of the cell attaching both kinetochores of the chromosome. So you want to be avoiding these situations. The situation that you want to have is this situation here. So the way that the cell solves this problem is that going from this situation to this, like this is a group of proteins that includes some of our favorite kinases, but I will not talk about it. But I will talk about how you make sure that this situation is the case. And the mechanism here is that as soon as the cell reaches this point, what happens here is that the APCC, the infamous APC complex, anaphase promoting complex, is activated, and also things happen with cohesin, condensing, kinases go down, and so on, and you can proceed to anaphase. While this is not the case, the spindle assembly checkpoint is silencing the anaphase promoting complex, and it's telling it, like, you know, don't go there yet because we have this situation here. The biology of this is known for more than a decade, actually, although details keep forthcoming. But the bottom line is this. The spindle assembly checkpoint somehow has to sense that you don't have attachment. And of course, there was a lot of literature about it, about the spindle assembly checkpoint sensing the attachment. There were, and there still are, a lot of theories about this happens. And this was the main problem that we had. How is the spindle assembly checkpoint machinery, don't think of it as a protein, but as a network of things, sense the lack of attachment? So a crucial protein in that case is the so-called MPS I kinase that you see over here. So MPS I, what it does, so here is a drawing of the kinetochore here. This is the so-called NBC-ADC complex. It's quite a few proteins. This is the KNL1 protein, and these are the two proteins that will bind to the microtubules. So now, what MPS I kinase does, it phosphorylates a lot of these substrates over here, MAD1, BAB1, BABR1, and when this is happening, when MPS I phosphorylation is present, these proteins stay on the KNL1. When they are not in KNL1, then the SAC, so when they're in KNL1, the SAC, the spindle assembly checkpoint, which these proteins participate to, is assembled. It silences the anaphase promoting complex, and these cannot, the APC cannot ubiquitinate different mitotic regulators. And MPS I is key for phosphorylating things in the NBC-ADC and in KNL1 to make sure that the spindle assembly checkpoint complex is assembled. So now when we go to the off state, the MPS I somehow stops to do the job. That's the bit that was quite unclear. Then microtubules get attached through the NBC-AD, the HEC1 and GLUF2, and the terminal end of KNL, and as soon as this happens, the BAB1 and BABR1 go away, the SAC gets it assembled, and the cell can proceed to anaphase. So, I'm drawing MPS I as two bits here. This bit is the kinase. It's a classical kinase domain. It uses ATP and phosphorylates protein. It's the business end. It's very interesting. We're still doing inhibitor structures for that. I'm not going to talk about it. But, you know, a kinase is a kinase. The question really, and that is the same, that's why I was working on polokinase, that's why I'm working on BAB1 and BABR1, is like, how do you tell the kinase to go to the right place at the right time? It is not, you know, you cannot regulate activity. You have ATP, you have substrate, well, you go and phosphorylate it. And most kinases actually are quite promiscuous. They have, when you put them in vitro, any of these kinases can happily phosphorylate very efficiently casein within minutes. It's a beautiful assay. But how do you tell it to be at the right place at the right time? And you know what we have figured out, and many other labs actually, is that there's regulatory domains that localize the kinase there. So that's a structure that we saw quite some time ago. We found out that there is a domain like that in MPS I, in BAB1, anti-BABR1, evolutionary conserved. We solved the structure and we looked at the structure, and this was one of the cases that we did not learn a lot of things. We tried to do histone hybrid screens, we tried to do mass spec screens. We knew, so the TPR is a typical interaction domain with other proteins or with lipids sometimes, but we could not figure out. We did extensive mutagenesis based on the structure, more than 20 mutants. We made stable lines of all these and so on. It was one of the cases that doing structures can be a tad disappointing. So the important experiment here actually, and this is a collaboration with the lab of Gert Kops in Utrecht, whose his main research focus is the cell biology of late mitotic events. Anyway, my postdoc went back to Gert, and what we did is this experiment here. So you look by standard microscopy techniques at the localization of MPS I in kinetochores. And you see at the wild type, you know, we normalize that 1. There is quite a bit of MPS I in the kinetochores. Then when we take out the first 60 residues that are not part of this interaction domain, you know, it is less MPS I. When we take out what we think it is based on our structural work was the interaction domain, well, you do get a significant difference, but it doesn't go to 0. MPS I is still there. When you take out all the first 200 amino acids, both this unstructured tail and the TPR domain, things go to 0. So that's what gave us the first clue that there is an interplay between the NT and the TPR domain, which actually depends on Aurora 2 action. And it's the synergy of these two domains that actually leads to the localization of MPS I. And knowing that, the next question is that where does it localize in the kinetochore? So one obvious hypothesis, well, one was the KNL, this was not the case. Another one was the NDC80 or the HEC1 protein, and specifically its N-terminal end that is in this part of the complex. So indeed, actually, if you do a few experiments here with RNAi knockdown, you can see that it is HEC1 when you get rid of HEC1. The RNAi experiments are here. You don't get localization anymore, and it's a phenotype quite when you reconstitute with recombinant HEC1 on the RNAi background, you see like when you reconstitute with a delta 207, you don't get colocalization in the kinetochores, while when you reconstitute with wild type, you get it. When you do GFP only, you don't get it. So very likely, at that point, we were dreaming of our cell paper 5 years ago. We said, okay, it is MPS I that interacts with HEC1, and that is the key of how MPS I goes at the right place at the right time. Of course, we sent the paper, and then we got a very kind letter back saying, okay, that's fantastic news, we'd love it. Can you show actually that this is true, that MPS I binds directly to HEC1? And at the time, actually, we were unable to show that. Somebody else was about to publish the structure, so we wrapped up our data, and we had this paper back in 2013. But that work has allowed us to formulate the hypothesis that this here is a direct interaction, and that could be a key for regulating proceeding to anaphase. So at that point, actually, somebody actually kindly gave us some money to do the work, so we could hire a postdoc to concentrate there. And I had this heretic idea that I'll hire an NMR spectroscopist. So what we did here is that we took MPS I and NDC80 broccoli, which is a protein, it's a complex of 2 proteins, and we call it broccoli because it looks like the stem, and the interacting domains are the leaves. And we thought, okay, what we will do is that we will label this protein with 15 nitrogen, and what happens then is that when we put the protein in the NMR tube and we collect the so-called HS2C spectrum, we get a lot of peaks. So each peak there, it's, if I would blow it up, you would see it's not a dot, it's a peak. It has an intensity and a shape. Is characteristic for every interaction of a nitrogen and a hydrogen in your protein. So what we expect to happen if this protein interacts with a bigger protein, what happens then in the NMR tube, if they're in complex, they stop going around very fast. It slows down simply because it's bigger. Plus, the new protein will mask a few of the nitrogens there. So we expect these peaks to disappear. And this rather trivial experiment in the NMR field, actually, it was very clear that we got peaks disappearing, we got other peaks broadening. It was very, very clear that these proteins interact. There's lots of control experiments, they don't aggregate, and so on and so forth. So, because this is, it gave us quite some confidence now to go and measure again our more trivial biophysics data. This, in that case, is microscale thermophoresis. It's a technique that's been criticized quite a bit, actually. We had this data that were a bit uncertain, but then NMR made sure for us that we are right. And we could measure this interaction with large error bars, but it gave us a KD of 3 micromolar. This is a lot of experiments and so on. So we have this evidence now that we have a direct interaction with MPS1 and NDC-ATC. The next experiment we did is this here. So what we do now is that we look again at the interaction, but we have like now the black line. It is just with the whole of MPS1 that is binding to this broccoli. And you see there is no binding here. Now what we do is that we phosphorylate MPS1 and do the same experiment again. And now all of a sudden, you see just by phosphorylating MPS1, we get binding. And we don't get binding of 10 micromolar. We get binding at the level of 150 nanomolar at this point. Which, of course, it was very exciting for us. And moreover is that if we do it even with a kinase domaining, we still get the same numbers, 210 nanomolar, 150, that's the same. So finally we could show a direct interaction that's phosphorylation dependent, and we could establish that. At this point we could also show that this interaction is mostly with HEC1 and not the whole complex is needed. And this allowed us to indeed formulate our model, and the question was like, could it be direct? We said like, could it be direct competition perhaps with a microtubules binding? And what happens is that simply when microtubules bind, NPS1 goes away and it cannot phosphorylate substrates anymore, which would be the simplest model. And indeed, it was correct. When we titrate microtubules in our experiment, we lose the binding, and the more microtubules we put, the less binding we have. So indeed, we saw at this point that very likely what happens is that when microtubules go and bind to the kinetochores, this antagonizes MPS1 binding, MPS1 binding goes away, and that gives the trigger. Obviously, we confirmed that in cells. Indeed, we look now at MPS1 in unattached microtubules, a lot of MPS1, laterally attached microtubules, much less, and unattached microtubules, very little. So very likely that model is correct. We went on back using an EM structure that another group had published a bit before. We did some mutagenesis. We create these multiple mutants, some directly antagonizing tubulin on HEC1, some on the periphery. And then we could see that actually, interesting, the peripheral mutants over here decrease by more than 1 order of magnitude the binding of MPS1. And interestingly, we could show that this binding was, these mutants still bind microtubules, but they don't bind MPS1. So there was some type of non-competitive binding. And indeed, we could show that as well when we make these mutants in cells, not in vitro. You see there's a dramatic increase in binding, which correlates. This is the wild type. This is the mutant that showed a weak phenotype in vitro. That's the mutant that showed the strong phenotype in vitro. That you totally lose localization of MPS1 when you mutate these sites, but you lose it without losing the ability of microtubules to attach. We also looked at this looking at mitotic exit. I will not explain the experiment because I'm running a bit out of time. But in general, like this is what happens when you have HEC1 wild type. The cells take a long time to exit mitosis because they have to check everything. That's what happens when you silence HEC1 completely, that's the red line. And if you do the add back with a mutant, it's the same as completely silencing HEC1. So that allows us to propose our model, which is like that. MPS1 localizes the kinetochores by binding to the NPC-ATC complex. This leads to the assembly of the sac, but it's a very dynamic interaction. It's on and off very quickly. It's not a static interaction. So at some point, the microtubules will get a chance to attach as they're growing. When they attach, MPS1 cannot go there because that's a much stronger interaction. The sac goes away, APC gets activated, ubiquitinates everything in the neighborhood, and you proceed to anaphase. That's all. I, of course, will only, well, not of course, but I will only thank, unfortunately, the people involved in the last bit of the story. Yoshi is then a Mars spectroscopist that we turned with crystallographer and cell biologist. Eleanor and Nassos did a lot of work for that work that we published last year. And Carlos, Spiros, and Vilko are the guys in Herr Kopp's lab that did the cell biology. And there's a few long-term ongoing collaborations and a few people that worked on that for a long time. And Renee and Hert are the long-term collaborators on that. So thanks a lot. I hope I did not get you too tired. And yeah, thank you again.
Okay. So questions, please.
From your model, it looks like the job of MPS1 is to set a delay circuit. So it's to phosphorylate and keep the anaphase from happening until you can get the microtubules to attach. Is that delay sufficient to get proper anaphase? In other words, would there be also a need for some force sensing structure to make sure that the microtubules not only have attached, but they've attached bipolarly correctly?
Well, the force attachment is certainly there to differentiate between unilateral attachment and bipolar attachment. So certainly there is a force sensing mechanism there. Certainly there are many other mechanisms that depend on kinases. This is part of this mechanism. The interesting thing is that MPS1 is evolutionarily very conserved. So lots of the other sensing mechanisms, they involve BABAR1, which is not there in yeast, for example, while this mechanism is also there in yeast. So we think that this is a very ancient mechanism to enter a bit your field. So it is definitely not the only mechanism, and I'm not trying to imply here that MPS1 is the only one needed. Certainly when you get rid of MPS1, you get lots and lots of errors. The same happens if you get rid of cohesin or condensin, or I can go on a long list, but MPS1 is definitely necessary. And yes, there is space for a force sensing mechanism, and there's almost definitely is an additional force sensing mechanism in the kinetic course.
So, going back to the question of causality here, I mean, MPS1, is it causally related to aneuploidy, or is it just the consequence of multiple genetic instabilities seen in many different cancers? For example, do you have direct evidence that MPS1 is directly mutated in an early oncogenic event in human cancer?
No. No. It's not a tumor suppressor in that sense. If you look at mutation studies in different cancers, you definitely get MPS1 mutants, but that's correlative still. And antimitotics is not the first line in cancer therapy. Antimitotics is when things start to go seriously wrong. It's very unlikely that it's causal.
but it's correlated.
It's definitely correlated with many cancers. Actually, it's if you go to any database and look at MPS1, more than 50% solid tumors, they will always have a defective MPS1.
But going to the medical point of view, is it a druggable kinase, you think?
There are kinases, so the drugs that target the kinase domain directly, there's phase 2 and now there's a phase 3 trial from Nervano, the guys in Italy, that there is an MPS1 drug that's very likely going to the clinic. These are the classic kinase inhibitors. There was an acquisition of a company that had an MPS1 inhibitor in colon, NTSRC, for 30 million euro recently. So there's the kinase, MPS1 kinase drugs. We have actually quite a few structures with these guys. It's definitely something that goes to the clinic. Success, we'll see soon. It looks good. What I want to do is target the localization of MPS1, try to develop protein-protein interaction inhibitors. And I've sent yesterday evening the rebuttal to this grant, so if we get it, we'll see.
That's more difficult. It's more difficult than working with small molecules, right?
Yeah, it's more interesting than working with kinase. You know, the kinase inhibitors, lots of the kinase, there are kinase inhibitors in the market now for several years. You have two problems. One is that we get resistance in almost all of them, pretty soon in most of them. And the second one, yes, everybody claims they are very specific, and sometimes they're very specific in vitro. It's not the same picture in vivo, necessarily. So the advantage of PPI inhibitors, which are still small molecules, we don't plan to, these are intracellular, we don't go for antibodies. We believe you can get small molecule inhibitors of PPIs. Other people agree with us. It's a very active field. If we can get these things, they're almost by definition more specific, and there is a good chance, because of the specificity, that you don't raise resistance so easily. The challenge is to make them work.
Okay, can I have another question? This is however not about MPS1, this is about this autotaxin in the first part of your talk, where you have developed this complex molecule as an inhibitor. Autotaxin does not go to the active site, but is close by. You said there are inhibitors already in the market, but they attack the active site?
Yes. So there are inhibitors that they're in the market, meaning you can buy them and put them in your cells to do studies. So there is one inhibitor that, there's two inhibitors that on phase 2 trials for fibrotic disease on autotaxin. And now there's companies that have very potent inhibitors that are trying to look at it in cancer models. But I didn't talk about the biology of autotaxin. It is quite interesting. So LPA signaling has to do a lot with cell migration. So what you're targeting there is metastasis. And the truth is that in metastasis, you don't really care if you have a very aggressive small molecule that stops it, because things are not great anyway. But it looks like autotaxin might be involved in early stages, so we believe there's still, it is a bit of the concept of the lab when we go to inhibitors. We don't want to go on anything on the classic active site inhibitors. There's my other project I didn't show, JBP1, which is for parasitic disease. We do exactly the same. Instead of targeting, so that's a DNA binding protein that has also enzymatic action in parasites. We don't want to target the enzyme, because the enzyme is a thymidine something. Thymidine something, you get everything. We want to target the DNA interaction. So if there is a theme in the lab, is that instead of targeting the enzymatic action, we try to target the interaction and get rid of the enzyme from going at the right place. So again, autotaxin, there's a couple of phase 2 things. A couple of companies are entering phase 1. One is doing it with an inhibitor we published, well, we didn't publish, we patented back in 2011, the HA135, which we published later. They developed it now to a lead compound, they go to phase 1, it's great, but they also see it's very toxic. So now we try to be a bit ahead and get that inhibitor. Actually, this inhibitor, because of the steroid, we are publishing, we're not patenting, so because steroids on the market, you tell to a company steroid, they're not happy. But at least we exemplify the concept, and Amira Pharmaceuticals seems to have a compound that works in a similar way, and I think they will be publishing it soon. Sorry for the long answers.
No? Other questions? So I think that